Okay.
So the sort of symptoms we've been looking at include a lot of cases that we may not explain even after people look for all the usual pathogens. So if you look in the literature, which varies a lot depending on the country you're looking at and how good they are at their diagnostic assays, you can find a whole range of unexplained diseases, particularly in diarrhea. Even in the United States, where they test for anything they can think of, a lot of cases still remain unexplained after we test for the bacteria, and all the usual viruses. Same for respiratory illnesses, and syphilitis is a big target for viral discovery because it's such a severe disease and there is such a high percentage of unexplained cases. There's a small fraction of hepatitis that varies from country to country that seems to be for which there is no etiological agent that has been identified. Chronic fatigue was there was a big push to look for viruses. I think people have sort of given up on that now. It's such a varied disease, and really people have looked hard for trying to find new viruses without success. Now, there's of course a big effort to look at eradicate polio, and in the process of doing that, the cases of acute flaccid paralysis are identified, which are not caused by polio. And there's of course other infections that can cause flaccid paralysis. But among these, there's still a large fraction that remain unexplained. Now, the biggest target, of course, are the autoimmune diseases and cancer. Now, the way we look for these viruses is we basically enrich for viral particles. If you just take a biological fluid and you extract all the nucleic acids, 99.9999% will be human genome and RNA. So the way we facilitate this discovery is we enrich for particles. And the first step we use is basically the old-fashioned virology. We filter the samples in order to remove bacteria and of course the much bigger eukaryotic cells. And viruses will go through the filter, especially the smaller viruses. Now, we may not be getting the so-called giant viruses like the mini viruses. And the technique we use, the next enrichment step we use is a nuclease treatment. So we take what goes through the filter, which is acellular, and we throw a big mixture of nucleases, that is DNAs and RNAs, to remove all the free-floating so-called naked nucleic acid, to basically get rid of them. We then extract, and what we extract is of course a mixture of the DNA and the RNA viruses and everything else which survived the treatment. Now, the key step here is called random RT-PCR. We use primers that have a tag, so we can pull multiple samples into what we sequence. But the key here is this: the randomized 3' ends, which means that theoretically these primers are non-specific, which they should amplify everything, all the nucleic acid present in your mixture. We do an RT step. RT also is a DNA polymerase, so it will also copy DNA strands after denaturation. We do a second, we come back to do the copy strands with a DNA polymerase, Kleno polymerase. And if you take that product, and then we do a PCR round to get a lot more DNA. We're using this primer, which is the same as the one for the RT and the Kleno step, about 30 to 40 rounds of PCR. If you take that product, if you run it on a gel, it's a smear. It's not you band for the PCR, it's a mixture of all these nucleic acids. Now, how do we know this technology works? Because we've been able to identify using this criteria, which is the expectation score, that what we find is not due to chance, all types of different viruses. DNA, RNA, single-strand, double-stranded, and all kinds of exotic and not so exotic samples. So the technique is fairly non-specific. And the ones we missed are probably because we just were not using the right kind of samples containing these types of viruses. But I'm sure if we had a coronavirus sample with a high enough titer, we would detect it. Now, what do we do with this sequence information? We don't look for DNA level homology, we look for protein level homology. That is, we take the sequence, the DNA sequence, and we translate it from both directions, both strands, every base, so base 1, 2, and 3. So we do all the possible reading frames of the DNA sequences into a protein. So for every sequence, we have 6 proteins. Then we take these protein sequences, and we use Blastex to look for homology. And homology, for example, when you compare related viruses, what you'll find is very conserved regions. And when you look at more divergent sequences, you can barely recognize the homology, but you can still find it. And so this is our pipeline, a bioinformatic pipeline. We take the sequence data, we remove the barcodes. Well, first we use the barcodes to bin the different patients into one run. We subtract some of the primers, we remove identical reads, we do some quality trimming, and then we do this de novo assembly. That is, we take the reads at the DNA level now, and we try to make a bigger contig. So rather than having, say, 200 or 300 nucleotides, if there is a high density or high concentration virus, we will be able to assemble the whole genome by overlapping with similar bases. If you have a rare or low titer virus, you only get a few reads and you do not form a contig. And then we take this information and we do this BLASTX. BLASTX is a basic alignment against GenBank, and you can use GenBank viruses only, which is the first thing we do, because it's a smaller database, and computationally it's much faster to take your sequence information and to look for homology against just the viruses. Then we get a long list of hits or high level similarity, and then we subtract from that long list. We take these blast hits and then we blast it against everything in NR. NR is the non-redundant part of GenBank, the unique sequences. And when we get a hit to NR that is a better hit to a virus that is a hit to, let's say, a bacteria or a fungus, we then remove it from the BLASTX virus hits. Because a lot of bacteria or phages, let's say, have things that distantly look like viruses. And we don't want to confuse those with real virus hits. And then we have our hits sorted on the website. So this is what it looks like when you have a very good hit. You see the protein alignment. This is our sequence translated from our contig of DNA, and this is a virus sequence in GenBank, in the viral database. And you can see this very good homology. And if you look at the expectation is sort of a mathematical way of giving you some statistics on the possibility that this is a chance event, given how big these databases can be. And here it's 0, which means there's really no chance that this would happen by chance. This is a more borderline hit, a more divergent virus relative to what we know. So the E-score is 0.003. And you can tell, if you look at this, that yeah, maybe not. You got levels of high conservations, which are usually for the shape of the protein, has to be more conserved. When we follow up on these more borderline hits, the first thing we do is we try, let's make this a bigger sequence. If you have a short sequence like this that gives you a borderline hit, if you're now using laboratory methods to sort of walk on the edge of that sequence, you're going to go from maybe a few hundred bases to, let's say, a few thousand bases. You redo the same alignment, you're going to see a much longer alignment, which is a much better E-score. You look at the gene organization, and you can tell whether this is the Callice virus, as was the case here, from an animal sample, from I guess a sea lion, versus sort of something else, maybe something unknown. Now, when we do this sort of experiment, we don't just find viruses to mammalian viruses. We find a whole bunch of interesting stuff. For example, in feces especially, we sometimes find viruses from protozoa. Presumably, these people are infected with Cryptosporidium along the virus. The virus is shed in the feces. We also found in pigs now, in pig feces, viruses from worms. Now, most pigs carry worms, and you're finding the viruses of their worms. And of course, we find a lot of bacterial phages. And here's, for example, is a fairly large phage that we get the entire genome. This was actually from a human. And what we're trying to do now is to go back and see whether the bacterial population in this individual is very clonal, which would explain why we had so much of this virus, as opposed to the usual mixture that you would expect in human feces. So, and the other thing we find a lot of in feces are plant viruses. So these are feces from people with diarrhea that had returned from a trip in Nepal, probably trying to climb the Himalayas, and they had diarrhea, and this is the sort of viruses that we found, sort of reflecting what these people ate. So there's a lot of misleading stuff, especially in feces. The other thing you can do is a little more complicated. We have a good collaborator that does that. You can look for things like ribozymes. Ribozymes are catalytic RNAs that can actually, functions like nuclease. And if you can look for patterns that are typically very conserved in ribozymes, and you can find them, and then people can then synthesize this RNA and actually show that they're catalytically active. So there's all kinds of goodie in the sequence information, but what we decided on a long time ago is to focus on the mammalian viruses. Now, this is more of a graphic representation of what we do. If you imagine this is a clade of viruses with all the different genotypes and species within, let's say, a viral genera. The closest new virus would be, the more related, the lower, that is, the better the E-score. And the more distantly related, the weaker the E-score. That is, the higher the E-score. But at some point, a virus is going to be so divergent that you will not recognize it as a virus, because the alignment will be such a bad score that you think, I don't know what it is, maybe it's something else. So you're not going to pay too much attention to it. So if you have a new virus here, you can now look around for its relative that you might have missed before, and now you're going to recognize a virus that was previously, say, invisible to your BLAST analysis. So simply adding sequence of viruses to the database, regardless of whether they're pathogenic or not, helps you discover other very divergent sequences of viruses. So here's some sort of practical applications of this technology. This is a study I did a few years ago looking at attenuated live vaccines. And the purpose was to basically convince people, look how clean your live vaccines are. So this is a shingles vaccine, which I highly recommend for people over 40, let's say. So we sequenced that. Of course, it's a very pure sort of solution. And we could resequence the entire herpes genome in that vaccine. Now notice these peaks. That means that the random RT-PCR is not perfectly random. It has favored regions where it binds. We're not quite sure why, but for our purpose, it's random enough to find these viruses. This is a polio vaccine, oral polio with the 3 genotypes, and again, we can resequence all 3 genotypes in this vaccine. So it was a very nice vaccine prep. And of course, you find some unidentified sequences. You also find some bacterial sequences in there, but at least there was no other virus. Now, the good thing is that the virus is concentrated enough. Not only can you sequence the genome, but you can look at the quasi-species of the virus. That is, if there is diversity, like for example HIV or Hep C, exists typically as a mixture of related sequences as it evolves very quickly in the patient. And if you look at the depth of sequencing, so for some region of the genome, we could sequence, like 8,000 reads over the same part of the genome. And if there is a minority population in there, you can actually detect it, because over some positions you'll have mixtures. You'll have an A and a T, 50%, 50%, or 1%, 99%. So you can look very at high resolution at the mixture of related sequences. And for example, the polio live vaccine is known occasionally or rarely to revert back to the pathogenic form. And what we could see, and we know what those mutations are. And there's one at the very beginning, the 5UTR untranslated region. And so we looked very carefully, did the vaccine already have some of these mutations? And the answer was no. And people already knew that when you revert to polio, it usually happens during the replication in the gut of the vaccinated person, and somebody else gets challenged and you get some revertant viruses. Now this was the one that got a lot of press because it was a bit more interesting. This is a Rotarix vaccine, and it's an extremely good and very safe vaccine. What we did find, though, is that the vaccine was contaminated with a pig virus, pig circovirus 1, which is a fairly harmless virus for pig. There's another one called pig circle virus 2, which is quite nasty for pigs, but this is harmless. There is absolutely no indication that this virus from pig actually replicates immunity. And when we looked at pork, we went to Safeway, we bought different pieces of pork. Most of the pork is infected, so people are eating this virus all the time. Nonetheless, it was in a vaccine that is given to infants, and the FDA did not like that at all. And but the vaccine nonetheless is so good, it is so important that they decided to tolerate this contaminants while the company slowly and very expensively sort of reformulates their vaccine to make it free of this contamination. But it is a harmless contamination. Now, where did it come from? It's likely that trypsin, which is used in pathoging these cells and actually making the cell line susceptible to rotavirus, comes from pigs, and it's likely that some of their trypsin was contaminated and contaminated their pipeline actually very early, so that even the vaccine they used for the trials, which were very successful trials, was already contaminated. So it's a contamination that's of no real consequence, but it's still a contamination. Now, why this turned out like this, we still don't know. It's likely that they had some kind of a PCR contamination or something bizarre. But so at least we cleaned up the literature, and people could ignore that virus, unless they're interested in viruses of the ocean. Now, another thing we've noticed, and now I'm changing field a little bit to more human physiology, if you want, is how common people are infected with viruses without being sick. Here we took a look at a very interesting set of sample we got from a guy who studied polios, and he had his 2 children, and he basically, the first year of their life, when they were being vaccinated by live polio, this is like 20 years ago, he took a stool sample from their kids and put it in the freezer. So that's quite a brave thing to do. And starting at month 3 until it's like almost weekly sampling from 2 of his kids. And the kids were vaccinated with polio. So you see polio here. The first, at about 3 months, there's polio, and polio lingered for a few months, and you could see some different genotypes of polio. Here's polio, the second inoculation. You only saw polio now, one of the genotypes, for a very short 1-week period. And by the third vaccination, oral vaccination, you could not even detect it. What you saw was another enterovirus. And the same for the other kid. So this is typically polio vaccination. The first challenge, you get the live attenuate, replicates for a little bit. The second one, less. And the third one, hardly at all. But what we found interesting was all the other viruses that we saw by PCR. So some Boca viruses, Picobi RNA viruses. This is the anelloviruses, which are like everybody has anelloviruses, typically in their blood. But very mysterious virus. And here we got adenoviruses. My point here is that these kids were healthy. There was nothing unusual about them. There was no hospitalization. They may have had the usual runny nose that kids that age get a lot. But the point is that pretty much at any time you're looking at a kid's species, it's going to be some kind of shedding of viruses. And for all we know, it's good for them. It prepares them, their immune system for the future. Another thing we looked at were little pigs on a high-density farm. So this is a great environment for virus transmission, obviously. And what we noticed now by metagenomic sequencing of their feces was the number of viruses shed in those pigs. Now there were pigs with diarrhea in blue and healthy little piglets. These were pigs, I think, just a few months old. Pigs don't live very long in a farm. And what was shocking was just the number of viruses that they were shedding. And even the healthy pigs. Now the pigs with diarrhea had just a few more viruses, but even the healthy ones were heavily infected. And my interpretation of this is that the maternal antibodies that pigs get was protecting them. And farmers will tell you that if a little pig does not suckle its mother, it will die very quickly. It's a nasty infection. So these maternal antibodies are extremely crucial, at least in this situation, keeping the pigs healthy. Now some of those viruses are very common and everybody gets them a lot, but some people are hurt very badly by these infections. Now, again, I think the reason for that, there are two reasons. Some people have very rare mutations in their innate system that makes them susceptible to just one type of virus, but not the others. And of course, the other reasons why what is usually a harmless infection can actually kill you is if you are immunodeficient, whether you're HIV, AIDS, or some more severe immunodeficiency. And people have been looking at this field. It's a very active field, getting people that are severely injured, and people are finding mutations, usually in innate response genes in these individuals. So while most people will have no severe consequences from these infections, those unlucky enough to have these very narrow susceptibilities can have. This is a bit of background on the parvovirus. And if you have a dog, you probably vaccinated your dog against parvo. Now, when we reported that, of course, first thing people do is they design primers, specifically this new parvo, and they start looking at their samples. And again, a bit of background. When we started this, the only parvovirus in human was B19, which people are probably quite familiar with. It causes usually a harmless infection in kids, but sometimes very severe sort of anemia type disease. So we found parvo 4, and a few years later, somebody found, actually the same year, somebody found the first human Boca viruses. Then we found a few more, because there are actually now two species of human Boca viruses. Then just recently we found still two more in fecal samples. So parvoviruses went from basically one species, and now we have 5 species of parvoviruses. So when people started looking more closely at this blood human infection, they started to find all kinds of animals-related viruses. There were primates, cows, pigs, and now there's even a bat, a parvovirus related to this new group. The next step typically is to develop serological assays and to express these capsid proteins in an insect cell system, and to do some serology. How many people are infected with this new virus, and what kind of people are infected? And what people realize is the people who had antibodies to this new parvovirus were people with blood exposure. So injection drug users, hemophiliacs, treated with coagulation factors, and a small fraction of men who have sex with men. Now, heterosexuals, very, very low rate of antibodies. So this is a blood-borne virus. And it may have sort of hitched a ride, for all we know, on the hepatitis C and the sort of HIV epidemic. This is another nice study done by people in England. They had longitudinal samples from hemophiliacs. They were sort of going back and looking at their HIV sort of infection rates. And what they realized is even with a heat-treated coagulation factor treatment, people were still seroconverting to this infection. And these parvoviruses are known to be really tough little viruses. That's why dogs get their own species of parvoviruses just from sniffing around the dirt from other dogs, from other dogs. So the big question now is it a pathogen? We're still not very clear. It may be that it's a pathogen only in the few people who have these rare genetic susceptibilities to parvoviruses, or to one type of parvoviruses, but not to everything else. Now, of course, it's likely that the people who are immunodeficient in a wider sense are quite susceptible. When we looked at healthy blood donors from Los Angeles by PCR of their blood, we found about 2% positive, which I don't know what that means. And that's probably the number of people who have blood contacts in LA. When we looked at like 4 hours. When we looked at now sort of kind of another group of individuals, these were more of those people with high risk for HIV, but RNA negative for HIV. We got 200 of those samples. There was a statistically higher rate of positive with this parvovirus. Now, does that mean that those people's symptoms, this fever and this sort of HIV acute infection-like syndrome, were caused by parvo 4? We still don't know, because it may simply reflect the fact that these guys have sort of the lifestyle that exposes them to more viruses in general. It doesn't mean that the symptoms that they had were caused by this virus. Now, there's been anecdotal cases of parvo-4 in other compartments of the body. For example, there were a case of encephalitis in India where the parvo-4 positive in cerebrospinal fluid. And there was also detection. Now, another use of this technology is to look at sort of the outbreaks. And this was a very interesting case that we got involved with of an hemorrhagic outbreak in Congo, DRC. And what happened is 2 teenagers basically died of an hemorrhagic disease, bleeding from just about everywhere. And when these outbreaks occur, people are very concerned about Ebola, of course. And Eric Leroy, who works in West Africa, tested these samples for all the usual suspects. And they were all negative for all the known hemorrhagic diseases. But he actually tested one guy because the 2 kids died too quickly. Nobody collected blood. But the third person was a healthcare worker. So there was fear of transmission within the hospital. And he survived, probably because he had better care. And we got his sample from the third person. And what we found was a new virus. But what was surprising was a rhabdovirus. And rhabdoviruses are not usually thought of as hemorrhagic disease viruses, unlike the known sort of hemorrhagic causing disease viruses. The only exception was rhabdoviruses here that cause an hemorrhagic disease in fish. So we're a bit surprised by that, but there is precedent for it. And for example, the related viruses to the human one are found in cattle. They don't cause hemorrhagic disease, but these viruses are transmitted by a little bug here. And the bug expert can recognize this. So of course we wanted to confirm that this infection found in the third guy was really real and that there was a seroconversion. So they because he survived, they managed to get blood from him a few weeks later, as well as contacts. So this is a healthcare worker, and you can clearly show using a pseudotype assay that one of my colleagues in San Francisco, Graham Simmons, developed, that he had antibodies to this virus. So, and now some of the contacts, other healthcare worker taking care of this healthcare worker, were negative except for one other one. And he was actually asymptomatic. So this virus is not always going to cause hemorrhagic disease, because in this case, this person also got infected, did not get sick. So we wanted to see a wider survey of the population. So we got hold of 50 blood samples from another part of the country, of Congo, and they were all negative. So our conclusion is this: there is probably some arthropod vector that infected the first 2 kids that died of this hemorrhagic shock disease, and one of them transmitted to this healthcare worker who survived but was in poor shape, and he actually transmitted to one of his healthcare worker but did not get sick. So what people are doing now is they're going back to the village and they're going back to other parts of West Africa to look at serology. Are other people seeing this virus? And they're trying to identify the insect vector. So they're going back and they're basically collecting bugs. They're bleeding the cows and the little rats and whatever they can find in this village. So far we haven't found the reservoir for that virus. And it could be it's going to be one of those infections that just pops up once, doesn't pop up again. But my gut feeling is if it's still circulating, we will see this virus again. Now, another field we've been doing a lot of work on is this acute flaccid paralysis. And for that, we've been looking at stool samples. And of course, the WHO program, together with their collaborators in the countries India is off that list now, are collecting stool samples from kids with non-acute flaccid paralysis, and they're testing them for polio. And when they don't find polio, they just put it in the freezer. And after a few years, they empty the freezer. So we got hold of some of those samples and started to look for viruses. And here's sort of a typical example And this is the number of viruses that we found in those species samples. These are not plant viruses, except for one here. These are mammalian viruses. And this was using 454 sequencing. So we're probably underestimating by a great amount the actual number of mammalian infections. So these are very poor kids in Pakistan who are exposed to a lot of contaminated food and environments. This kid here had 5 viruses, 2 picornaviruses, 3 picornaviruses, actually 4 picornaviruses, a rhinovirus, and also we found a plant virus. And this kid died actually. But this again is an example of how children in developing countries are just constantly infected by. Example of picornaviruses, polio is a typical example. When we started this study, there were basically 4 groups of the picornaviruses that were known to infect you. And enteroviruses, this is where polio and rhinoviruses are in here. People are very familiar with par-eco, it can cause encephalitis, it can cause a liver problem. This is more of a diarrhea virus, Aichi virus is in here. Since we started this, we added 3 more groups of viruses, 3 more genera in the Picornaviridae. This we showed was related to also diarrhea. Cosavirus is, I think for me, the most interesting one. It's a very, very common infection, very diverse group of viruses, just like the enteroviruses. And then cardioviruses, the first one was found by one of my old postdoc when he moved to his job in California Public Health. And so I'm going to go a little bit more detail, especially on this group. What was interesting, when we started sequencing these amplicons, because now we have the first hit by deep sequencing, we can start using PCR. It's cheaper and faster, and we start looking at a lot more samples, and we sequence our PCR amplicons. What we found was, like I mentioned, all these different genotypes and species of viruses. So it's quite likely that just like the enteroviruses, which are mostly harmless, but there are, if you want, subtypes of it, like polio, that are extremely nasty. And Entero 71, for example, which is quite nasty as well. So my gut feeling is that some of these viruses will be pathogenic in some cases, and most of them will not. When we did PCR, just to show you how frequent these infections are, if you look at sort of degenerate primers for enteroviruses, 75% of any stool sample at any time from one of those kids which were younger than 15 years of age with acute flaccid paralysis was positive with one enterovirus or another. Healthy controls were just about as equally, prevalence of infection was about as equally high. And the cosaviruses were also very high. There was really not a very big differences between the flaccid paralysis cases and the healthy control. So my point is here that cosaviruses may be the second most common human enteric infection after the enteroviruses. And again, just look at the healthy kids, just how commonly people shed these viruses. The problem with this diversity is it's not just one species, it's 5 or more species with all the genotypes within these. So when you do disease association study, you have to deal with this diversity. You can't just call them all cosavirus. You have to say cosavirus, A/23. That might be pathogenic, but A/25 may be non-pathogenic. So you have to start looking very carefully at the diversity of these viruses in your cohort. Another virus, so the cardiovirus we found, that my postdocs first found, and we found a whole lot more once we had that genome information, used to be called cardioviruses. They used to be only really known to infect animals, pork and mice. There was the occasional virus infection in human samples, but people always suspected contamination because they occurred after multiple passage in labs that typically also studied animal cardioviruses. The first one was found by deep sequencing by Morris Jones at the California Department of Health from an old sample that had been in the freezer for 30 years from a supernatants showing cytopathic effect, which is the sort of classical way of showing enterovirus infection. But they tried to type this supernatants, and they didn't know what it was. It did not react to their antibodies. So they just put it in the freezer. 30 years later, we pull it out, Morris pulls it out, and found the first virus, which was genotype 1. And he gets to name it. after his grandfather, actually, Saffold virus. But now we had that sequence by PCR. We started looking at our collection of stool samples, and we found a whole bunch, again, of a very diversified group of viruses. And it's quite likely that some will be pathogenic in some infections. People have started to do serology against some of those genotypes and found that, for example, even in Europe, 80% of people are positive for just 2 of those genotypes. So these are not just developing countries, they're also in Europe and in the US. In fact, we've been looking at sewage from different cities in the West Coast and the East Coast. We also find these viruses, so they're global, if you want. A collaborator of ours, Charles Chu, also at UCSF, had very good samples. They were longitudinally collected from an outbreak within a family of diarrhea. So he had the blood during the acute symptoms of diarrhea, and he had another blood sample collected a few weeks later. He was able to grow the virus, one of those cardiovirus, and what he found is that the virus could grow quite well in the presence of acute plasma. But when he took plasma a few weeks later, he could neutralize this virus. So not only could he put the virus in the right sample at the right time, but he could show that this was the first time this person had seen this virus because they developed antibody soon thereafter. So this is another way you can make a case that the virus you find is actually causing the disease. If you can grow it, if you can PCR it, if you can show seroconversion, and if you can show there's nothing else that might have caused this diarrhea. So I want to move maybe a little bit here now from, I don't want to just go through a list of viruses like this, but we've been doing a lot of nice work with animal samples, because frankly, if you want to find viruses quickly, just look at animal samples. There's just a whole universe out there of viruses. A very interesting case we did with our collaborators at UC Davis. Here she did not use deep sequencing, she used consensus PCR. She had what she thought was sort of an outbreak of brain cancer in raccoons. But there were several cases that came in, and raccoons had never been reported with sort of a brain tumor. I think the doctors will tell me which one is the tumor. So using consensus PCR, which is basically generate primers, trying to target all the known species of polyoma, she found a new polyoma. And you put it on a tree, had a very deep branch. And what was nice is that she then got a whole bunch of these raccoons that were behaving oddly, and they just dropped dead. She got 10 of those cases with glioblastoma, and all 10 of them were PCR positive for this virus. And a control group of raccoons, killed by cars, were all negative by PCR. So this is again a nice sort of a case control, if you want, trying to link this infection with in this case, a brain cancer. Polyoma, of course, are known to cause cancer. SB40, you probably learned in grad school, causes cancers in rodents. The next thing you can do, of course, is to develop antibodies to the capsid protein and showing that the virus is expressed in the tumor. So now what she's trying to do is basically grow the virus and inoculate raccoons to really prove, you can do all kinds of more interesting things with animals, that this polyoma actually can induce this brain tumor. Another interesting case we got, also from UC Davis, was a dead cow that neurological symptoms. Now, people get very worried when cows behave strangely because of prion. So they put them through all kinds of tests, and these all came back negative for the usual viruses associated with neurological symptoms in cows. So we got some of this brain sample, and we did this metagenomic, and what we found was a new astrovirus. Now, astroviruses are not typically thought of as anything else but enteric infection, occasionally causing diarrhea, especially in AIDS patients. But this was a new astrovirus. And then what we liked about it is that we had several cows. We had 30 cows with these sort of symptoms, and 10% of them were PCR positive for this virus in the brain tissue. So again, it's a bit like the raccoon. We had a lot of these animals with these symptoms, and we could show repeated detection of this virus. As a quick aside, There's a whole bunch of human astroviruses and animal astroviruses, other species infecting the cows. But there's other cases. For example, here is an astrovirus here that is typically causing diarrhea or nothing. That was found in there was a kid with encephalitis, and they found, but it was an immunodeficient kid. And they also found that the virus was infecting this kid's brain. So there was precedence. And this is an epidemic virus in mink farms where they start shaking and they have neurological symptoms. Again, astrovirus brain infection. So viruses that people usually think of as either harmless or typically harmless or maybe causing diarrhea, on rare occasions can cause much more severe disease. And again, here we got in situ hybridization with the RNA sequence, and we could show detection of this RNA from this virus in neurons in the brain of these cows. And this is the last case of a virus I'm going to mention here. These are dogs with vasculitis, and I guess this is what vasculitis looks like. And what we found here was the second case of these circovirus. Remember the circovirus in the vaccine that's also infecting pigs? We found another one infecting dogs. And actually this dog was actually co-infected with the bocavirus, one of those parvovirus. And these viruses came not from the feces, but came from the tissue itself. So when you find a virus in the tissue, you're much more confident that it's actually infecting the animal, and not just something that the animal ate. What we did with Pat Pisamento is in situ hybridization again, and we could find viral DNA from a replication within the cells in the lymph node of this animal with vasculitis. We developed an antibody using peptides and shooting peptides into rabbits, and we can show expression again here in macrophage. And if we're looking by EM in some of those tissues, you can find these inclusion bodies, which are basically packs of viruses of the right shape for a circovirus. So the evidence was pretty clear that this might be a pathogen causing vasculitis. We've been working with a company that does diagnostics for animals. They get a lot of fecal samples from dogs with diarrhea. And this virus is actually all over, and I think there's now reports of in Europe. So this is probably this might be a dog pathogen, just like the pig circoviruses. That's global. And of course, people love their dogs. As soon as this was published, people started saying, "Oh, should I vaccinate my dog?" Well, we're not sure yet, because ultimately, I think the proof of pathogenicity would be inoculation of some dogs to show that a disease can be induced, whether it's vasculitis or diarrhea or something else. So to wrap it up, we've been finding a lot of viruses since this sort of metagenomic approach started. A lot of new parvoviruses. We found quite a few of those. New picornaviruses. There were 4 genera, and now there were 3 more added. The astroviruses, we found 3 more. There used to be only 1. And the polyomas have been very active. There's actually one that was just published this week in Journal of Infectious Disease by another group. So there's an explosion of new human viruses. A lot of them are orphan viruses. We're not quite sure what they're doing. It's going to take some time for the epidemiology to catch up with the metagenomics. So, we're finding a lot of viruses. Many are orphan. We're not sure what they do, if anything, in most cases. There's a high diversity in some of those clades, which makes disease association much more complicated. You just need bigger numbers. I'm not talking about this very much, but we see phylogenetic evidence of cross-species jumps. That is, the trees of the viruses from one species do not all cluster together, all the humans and all the cows and all the bats. But you find bats infected with viruses from all these clades, and the same for humans. So we interpret this as cross-species jumping. And a lot of high rate of infection in healthy, young, as well as sick animals and humans. So if people are interested in this sort of approach, what's sort of the practical steps? Well, look for unexplained symptoms, that kind of could be infectious. And for example, pathologists are really good at looking at tissues and saying, yeah, that looks infectious. It looks like immune response is going on. Inclusion bodies, they're very good at sort of spotting things that may be infectious. You want to exclude the usual suspects. So you don't want to rediscover rhinovirus or influenza. You want to find something new. So you want to exclude those more traditional viruses. Timing, I think, is quite important. You want to sample early after the onset of the symptoms and the fever. If you can, it's better to have longitudinal samples. This way you can look at antibody seroconversion and confirm that this infection was recent and associated in time with the symptoms. Tissues are always nice. Of course, you get a lot more tissues from animals and from humans, but you can also look at plasma, feces, nasal swab, respiratory swabs. And of course, you're going to take, if you have tissues, you want a tissue with showing the pathology, like hepatitis, liver. Neurological symptoms, you prefer some kind of brain tissue. And the other type of tissues we like a lot are outbreaks, like this rhabdovirus in Africa. When suddenly a lot of people start getting these unusual symptoms, that's when you're likely to collect samples where you can make a better case that the symptoms are associated with the virus. Because you have, let's say, 20 people suddenly went to the same hotel and all got diarrhea. You might find noroviruses, but if you find something else, then you've got access to all these other individuals and you can see whether they all got the same virus. So again, some more practical steps. If you find a virus, the first thing you want to do is you want to exclude contamination. You want to make sure it's not a plasmid somebody had in the lab or comes from the reagents like the Chiagen columns. You want to do, if you can, a case control study where you have 100 people with, say, unexplained encephalitis and 100 people without encephalitis, where you both got the same tissue, CSF, for example. Because then you can do, what percentage of people have the virus? Is there statistically more in cases than control? You would like to see seroconversion. You'd like to see viral RNA or protein expression at the site of pathology. You want to use metagenomics because the other thing that people are starting to do now is to say, look, we looked for all the viruses and we only found one. We didn't find anything else because this technology now allows you to basically sequence everything that's there. So people will make a case by exclusion. Look, there's only one here, so it's probably causing this disease. You want to, of course, wait for independent studies to reproduce your data. And ultimately, you'd like to try some animal inoculation, which are, of course, much possible animal studies. So conventional PCR, very sensitive, very quick, very cheap, but you're only looking at what you already know. Metagenomic, basically throw a grenade, everything comes out, and you have to sort of wait with it and see what's likely to be important and what is not. And it's quite easy to jump to conclusions too early. So again, my colleagues from BSRI are listed here. This is sort of ex-members who contributed a huge amount. And these are great collaborators, the vets from UC Davis, UCSF, are all bioinformaticians. Bioinformaticians are key to this work. You really need good IT people to help you deal with the huge amount of data. Our friends from WHO polio eradication groups and Metabiota who got the samples, who are getting lots of interesting samples. Thank you very much.